improve the functioning and quality of life for all individuals on the spectrum. First of all, I wish to thank our sponsors, research institutions, Caltech, UC Davis Mind Institute, Stiles Nicholson Brain Institute of Florida Atlantic University, the O'Sullivan Foundation for continued support, and corporate sponsors, Charm Health and EdSite. Thank you very much. Okay. I'm having trouble with the screen share. Sorry about that.
Try clicking on the slides once you share screen. So.
Yeah, I don't know. I've had this problem since the new date was installed. Our community partner organizations are the Autism Research Consortium. They're here live streaming this event to their community. Thank you very much. And Thinking Autism UK. Our main goal is to enable translational research. We bring clinicians, researchers, and the community to this conference, Synchrony, every year. And we put out a request for study proposals, and we discuss the updates from the research every year at Synchrony. We also invite donors and investors to come to this conference to support startups in industry and also researchers. In 2021, we supported research across 10 institutions, and we covered broad areas of basic research, biomarkers, and treatment studies. We put out a request for grants in the first quarter, and after rigorous peer review process and meeting of the Scientific Advisory Board, we ranked these proposals by various criteria, and we made the grants for these proposals. I'd also like to thank our foundation partner, the O'Sullivan Foundation. Sean, if you're here, thank you so much. As a result of their matching grants, we were able to fund a lot more research than we otherwise could have. So these are our grants from 2020. Some of them are continuing grants this year. And these are our new awardees from 2021. I will not go through the details because most of them, all of them will be presenting today, and you can learn about their research and the updates this year. So here we are at Synchrony 2021 today, and tomorrow you will listen to the best and the brightest in this field, both industry and academia. So we will have 21 speakers from research institutions and 10 industry leaders. We will cover a broad range of topics from basic science and animal models, immunologic neurometabolic studies, microbiome, new therapeutics, clinical trials, and biomarker discovery. So this year's goals, again, are to fund over 1 million in research grants for studies, renewing the continuing grants from last year and some new ones. And we hope to present a research conference for publishing the new results. We also plan to have in 2022 a number of shorter meetings. One will be on biomarker research and development because it is crucial. We plan to have a roundtable to discuss case reports of patients with specific characteristics, specific subgroups, like adults with relapsing and remitting symptoms. And we hope to do a working group session to get case reports published, and also another workshop to take research from labs to FDA approval, and the challenges in that path. So those are the plans for next year. If you are a researcher, please join us to make a difference in the lives of these individuals, and also to be inspired by the best scientists and mentors. You will enjoy the support of our community. If you are a parent or an individual with neurological disorders, please support medical research. volunteer, fundraise, and join clinical trials. Any age is not too late, and even an improvement of 5% makes a difference. Our hope is for independence and happy, productive lives for all. I'd like to end with a quote for our caregivers who are joining us today from Viktor Frankl's book, Man's Search for Meaning. At the heart of Frankl's theory is a conviction that the heart of human drive is not pleasure, but rather the discovery and pursuit of what the individual finds meaningful. We must never forget that we may also find meaning in life, even when confronted with a hopeless situation. For what then matters is to bear witness to the uniquely human potential at its best, which is to transform a personal tragedy to a triumph, to turn one's predicament into a human achievement. And what we wish to achieve is improved mental health, physical health, improved functioning, quality of life, and independence for all. We may not see the finish line yet, but we are making a difference, and the best minds are here with us today. Welcome to Synchrony 2021 from all of us here at the Brain Foundation. Thank you very much. So I will start the morning session with an introduction to Dr. Richard Frye, who's going to be chairing the session. Dr. Richard Frye will be speaking about an update on mitochondrial research. He's a child neurologist with expertise in neurodevelopmental and neurometabolic disorders. He received an MD and PhD in physiology and biophysics from Georgetown University and completed his child neurology residency and fellowship in behavioral neurology and learning disabilities at Harvard University Children's Hospital, Boston. He has authored over 150 peer-reviewed publications and book chapters and serves on several editorial boards. He has conducted several clinical trials demonstrating the efficacy of safe and novel treatments that target underlying physiological abnormalities in children with ASD. He is the chief of neurodevelopmental disorders at the Barrow Neurological Institute at Phoenix Children's Hospital. I'd like to mention that he's on our scientific advisory board and extremely helpful to our foundation. Anytime I had anything that I needed help with, he's always right there to help. So thank you so much, Dr. Frye. Please take it away.
Thank you so much for that wonderful introduction. And I'm glad to see so many people here and so much interest. So today, I will talk about mitochondrial dysfunction in autism and the roads to understanding it and treating the mitochondrial dysfunction to improve symptoms in autism. Just some disclosures. I have funding from many places, including the Brain Foundation, and also are doing industry clinical trials in our advisory boards. I'll just say a quick disclaimer I have in every talk is that the treatments that we talk about many times, and for the most part for autism, are now not FDA approved treatments. That doesn't mean that they can't be used, but it means that we have limited evidence at this time, and that it's important when using treatments to consult a medical professional who's knowledgeable about the risks and benefits of any type of treatment that you would use, both off-label and on-label. So, today we're going to talk about the mitochondria. The mitochondria is a very important part of almost every cell in your body. It's best known for making energy. We have anywhere from hundreds to tens of thousands of mitochondria in each cell of your body. And it's a very complex part of the cell. It actually has two membranes to enclose the machinery. It has these little folds in the inner membrane called cristae, where a lot of the work happens. It actually has some of its own DNA. It's the only other part of the cell that has its own DNA besides the nucleus. So it's very complicated because some of its functions are regulated by the genes that are specific to the mitochondria and not on the chromosomes. And so we have our normal mitochondria, which has lots of power, gives us energy. And then what we're thinking is that sometimes there's dysfunctional mitochondria where, it doesn't seem to be working that well. It doesn't have the power to do what it needs to do. And what we're learning is that this is not just a circumscribed, what we call mitochondrial disease, but that the mitochondria being central to the many of the cellular processes is actually dysfunctional in many disorders, since it's so important to support cellular function. Really, we're still learning about mitochondrial disease and dysfunction. Mitochondrial disease really was first described in 1988. The first papers were published on the first mitochondrial diseases in Science and Nature, two really good journals, of course. And so when we think about it, in the time span of medicine, 1988 is pretty much a blink of the eye. So that's why, we're still learning about mitochondrial disease and dysfunction. And one thing we know is that when there is a problem with the mitochondria, we see that there's symptoms within the high energy dependent tissues of the body. And these include most usually the nervous system, so neurologic disease, the GI system, so gastrointestinal disease, and the immune system. And when you think of the comorbidities that we see in kids with autism, these are the three areas of the body that are most affected. So it's no wonder why, the mitochondria seems to be involved possibly in autism. And the other important thing to know is that the mitochondria is not just the powerhouse of the cell, but it's important for other functions, including regulating cell death and oxygen radical regulation. Really, the evidence for mitochondrial disease and dysfunction is growing. And probably, I think one of the best reviews was the paper that Dr. Rosignol and I published in 2012. We were kind of interested. We had heard that there's more and more evidence that there might be some involvement of the mitochondria with autism. So we did something called a systematic review and meta-analysis to see what the evidence was. And we found some very interesting findings. Our findings were that the papers that were published, there was 3 papers to look at very strictly defined mitochondrial disease, which is a very particular specific diagnosis. And we found that studies that looked at that found that about 5% of children with autism have mitochondrial disease, which is a very severe form of mitochondrial dysfunction. But when we looked at studies that looked at biomarkers that the mitochondria wasn't working, that is elevated lactate or pyruvate or lactate to pyruvate ratio and such, we found those rates were much higher, somewhere around 10 to 30%. And so what that suggested to us is that there was something going on with the mitochondria, that is, the mitochondria wasn't working, but it may not fall into that category of mitochondrial disease. So they might have been this broader sense of mitochondrial dysfunction. And so, and that's not really, surprising in a lot of ways, because one of the things we know is that the mitochondria is affected by environmental factors, both the external environment and the internal environment. So the external environment, like toxins, like the microbiome, the microbiome technically is outside your body, even though it's in your gut, but also things internally. Oxidative stress in the body and inflammation will affect how the mitochondria functions. And we know these are all involved in autism. So it's not surprising that the mitochondria would be affected. But it's important to know that when the mitochondria isn't working, there's consequences, including increased oxidative stress and inflammation, autoimmune disease, immunodeficiency, neuronal dysfunction, problems with neuroplasticity, cerebral folate dysfunction, autonomic dysfunction, and gastrointestinal dysfunction. And what's important about the mitochondria is, again, it's at the center of many of the important cellular processes, many diseases. And it can, when it's not functioning correctly, it can start a vicious cycle, which can cause more dysfunction. And this is just one diagram that shows, well, dysfunctional mitochondria can increase oxidative stress, which can cause inflammation, which can cause more mitochondrial dysfunction. It can also deplete glutathione, which is important for mitochondria to work, or actually create mutations in mitochondrial DNA. So the mitochondria may be very central to a lot of the physiological abnormalities we see in autism. This is one paper we've published recently. This is a diagram from there where we looked at the prenatal factors that could affect the mitochondria. And here you see on the left a list of prenatal factors that have been linked to autism, and how they either negatively, that is in orange, or positively, which is in green, can affect mitochondrial function. So there's a lot of evidence that many of the prenatal factors that are linked to autism could act by affecting the mitochondria prenatally. And we have some evidence of that also. And of course, on the right, we see mitochondria is important for many important pathways in the cell. I'm not going to be able to talk about the evidence that we found in the papers that we've published, but suffice it to say, we have a series of papers where we've looked at prenatal environmental influences, both air pollution, prenatal exposure to air pollution, and prenatal exposure to metals, both nutritional and toxic metals, to see how that affects long-term mitochondrial function. And we were actually able to show that that the mitochondria is modulated long-term, that in childhood you can actually measure mitochondrial function and link it to those prenatal exposures, suggesting those prenatal exposures may have actually formed the physiology of the mitochondria, have influenced the physiology of the mitochondria early on, and that these differences, these changes, were different between those that have regressive autism and non-regressive autism. The other thing that we have been able to show is that indeed there is a unique type of mitochondrial dysfunction in autism, particularly those with neurodevelopmental regression. And this is a recent paper we published with Andy Zimmerman, who was originally at Kennedy Krieger and then Harvard and UMass, where we were able to take samples from patients that he studied with and without neurodevelopmental regression, with autism, to show that those that have neurodevelopmental regression in autism actually have very active mitochondria that where their respiratory rates are much higher than normal. And this is important because usually when we think of mitochondrial disease, we think the mitochondria is not working well, so the respiratory rates are very low. Here we've actually linked a subgroup of autism to extremely high respiratory rates of mitochondria. So we'll talk about some of the grants from the Brain Foundation that we've received that is helping us find treatments and learn more about the mitochondria. This is one grant where we've tried to just simply take a mitochondrial supplement that should improve mitochondrial function, and very critically look at it through a double-blind placebo-controlled clinical trial. So there's a controversy about whether you should treat mitochondrial dysfunction or whether it has to be mitochondrial disease, whether you need a particular diagnosis of mitochondrial disease. And this is a nice paper with my colleagues Dmitry Nizov and Steve Kaler, where we argue and we give vignettes of diseases where you see actually mitochondrial dysfunction secondarily. And it's clearly not a disease of the mitochondria, but the mitochondria is affected. And we discuss how treating and to support the mitochondria, even though it's not the primary part of the disease or the primary cause of the disease, can actually improve function. How do you treat the mitochondria? Well, there's many different ways, but suffice it to say there are certain supports with certain specific vitamins, many of them B vitamins, but also CoQ10, creatine, certain folates, and support for oxidative stress. I'm not going to go through them all. But there are certain vitamins, and in our study, we use the NeuroNeeds vitamin, which is actually designed to support mitochondrial function, along with the Q-Needs vitamin, which is a vitamin kind of cocktail to support mitochondrial function. We developed a double-blind placebo-controlled crossover trial where first we enter individuals that have some type of mitochondrial dysfunction, as measured by non-invasively with the buccal swab technique, and then treat them with either the actual supplement or placebo, and measure how their mitochondrial function changes and how their symptoms change over time. This is an FDA regulated trial. We asked the FDA and filed for an IND, not to approve this as a drug at this time, but to increase kind of the level and of quality of the trial. And we had two aims. Our primary aim was to determine if this supplement could improve mitochondrial activity in 50 individuals with autism and abnormalities in mitochondrial function, and two, to see if it improved the core and associated autism symptoms. So what came out of it? Well, all these things are a process and a learning experience. So, of course, getting FDA approval, one of the things that we found is that we needed a certificate of analysis of the investigational product. And when you have a combination product like this, and this is why I don't think drug companies like to make combinational products, it's very tough to actually investigate analyze the product once it's mixed. And there is a lot of different issues, and certain chemicals interfere with other chemicals, certain substances interfere with others when you're measuring these things. So it took us about a year to find a company that could do this with enough precision and within a reasonable price point, because we had price quotes anywhere from $5,000 to $2.5 million. And we were able to do that. And then we had to produce a placebo that tasted identical to the product. And that took about a year. We had to do taste testing and blinded taste testing to actually develop the product. Of course, we had to advertise and recruit for the study. And then we had to find the participants, which is a little bit slower than we expected, which I'll discuss in a second. And then one of the major issues we found is the tolerating of the treatment. And it's about 60%. And I don't think this, in our experience, this isn't really specific to this product, but to treatments in general in kiddos with autism because of their particular aversions to certain tastes and textures. And really tells us that some of the ways that we've delivered products as far as pills and powders and such, and liquids and such, may not be optimal, and we have to, there's probably a whole area of research we have to look into of how to develop products with better delivery systems. We in the study we had 29 that were eligible, and in order to be eligible, after in screening, you need to actually have confirmed mitochondrial dysfunction. And unfortunately, we were hoping to get those tests back with a 2-week turnaround time, and it's been a 3-month turnaround time. So that slowed down entering people into the study. And we've lost a couple to follow up because they got tired of waiting. So it's new technology, and it's unfortunate that it takes time, but we're moving ahead, and we now have 3 completed, but we have definitely some that are going through that are in part 1 and part 2, and others waiting to enter the trial. So taking a little bit longer than we had expected, but I think we're learning a lot, and we'll get some really good information from it. The second, approach we're taking is by looking at in vitro cellular models. We started out this research many years ago using lymphoblastoid cell lines, and now we've moved into using fibroblasts to confirm and further our studies that we had in the lymphoblastoid cell lines. We use something called the seahorse. The seahorse is a instrument that has really revolutionized looking at mitochondrial function. It used to be that you could only measure 1 or 2 samples at a time of mitochondrial function. You had to isolate the mitochondria, which is a bit of a chore. And so the seahorse was developed about 10 years ago, and it allows you to measure mitochondrial function in living tissue, so in cell culture, in a 96-well plate. So you can put in replicates, you can manipulate the cell lines, and you can put both control and your experimental cell line on the same plate, which gives you a very high throughput and allows you to measure a lot of things. And so one of the manipulations that we decided to do when we started this was to see how, not just how the mitochondria was working, but how the mitochondria was resilient to stressors, particularly oxidative stress. So we developed an assay where we can use something called DMNQ, which increases superoxide in the cell, so that we can see how the mitochondria can actually handle stress. From this assay, we measure several measures of mitochondrial respiration, including what we call ATP-linked respiration, which measures how much ATP or energy the mitochondria is making, proton-linked respiration, which is very important. And this is actually a type of respiration that controls oxidative stress. And it does not make ATP or any energy. So it decreases the efficiency of the mitochondria. And then we can measure what we call the maximal respiratory capacity, how much the mitochondria can function, how high its respiratory rate can go. And then I take this difference between the basal respiration and the maximal respiration, which tells us about the reserve of the mitochondria, how much reserve the mitochondria has if it has to actually increase its respiratory rates due to stress. We verified, so again, we did this assay where we used DMNQ to cause stress, and this is just an assay where we verified that actually the mitochondria was being stressed. The red color you can see, which progressively gets more and more as we add more DMNQ, is a mitosox red, which measures mitochondrial superoxide, so oxidative stress within the mitochondria, to verify that our assay is doing what we think it's doing. We've done several experiments on what we call lymphoblastoid cell lines, which are immune cells that have been immortalized. And what we showed, and this is one study that looked at patients with autism, and they're typically developing siblings as well as unrelated controls. And what we did show is that the respiratory rates were much higher in the kiddos with autism as compared to their typically developing siblings, and the unrelated controls. And one thing we did show that's not in this slide is that that the siblings tend to have high levels of oxidative stress also in their cells. So the thing that differentiates the siblings and the kids with ASD or the is the abnormal mitochondrial function. We also showed that that if you divided up the kiddos with autism into those that had worse and better repetitive behaviors when these cell lines were harvested, or when the blood was actually taken from the child several years before these studies were done, that we could actually differentiate which children were more severe by measuring their mitochondrial function. In larger samples of lymphoblastoid cell lines, what we've been able to do is show that there's these two subgroups. There's a subgroup of children with autism who have normal mitochondrial function, and then there's a subgroup that have these very high respiratory rates, which, you know, I had previously shown you. We've linked to neurodevelopmental regression. What we showed was that not only do they have these very high respiratory rates at baseline, but when you stress these cells with DMNQ, these respiratory rates plummet much faster in the kids with autism than in the controls, particularly those that have these high respiratory rates at baseline, suggesting that there's this subgroup of kids with autism with a unique type of mitochondrial dysfunction who are very vulnerable to physiological stressors. And we've looked at some of the reasons why molecularly. In one study, we were able to link this to a protein called UCP2, which regulates oxidative stress at the inner mitochondrial membrane, and showed that this protein was upregulated in the subset of lymphoblastoid cell lines from the kids with high respiratory rates and autism. We've also linked this to changes in gene regulation, where we've found that the kiddos with high respiratory rates fail to upregulate important genes that combat stress, physiological stress in the cell, including PGC1 alpha, PINK1, MFN2, SIRT3, SIRT1, and so on. We were also showed that this might be related to regulation of mTOR, and that low-dose rapamycin may be able to correct some of these changes. And in one in vitro study, we showed that by using low-dose rapamycin, we could actually normalize the respiratory rates of these cell lines. So now we're actually moving on from the lymphoblastoid cell lines to fibroblasts to confirm these same findings in a different tissue, to make sure that this isn't, you know, just something because of something particular to lymphoblastoid cell lines. And then we're taking this a little further. And in this study, which was a collaboration with the French, we were able to use confocal microscopy to look at mitochondrial morphology, and actually showing that the shape of the mitochondria is different in cell lines that have these high respiratory rates versus the normal respiratory rates. That is that the cell lines with high respiratory rates had more complex mitochondria that were clustered together and closer to the nucleus, whereas the ones with normal respiratory rates had much longer, more evenly distributed mitochondria in the cell. And what we're doing now with the grant from the Brain Foundation is taking treatments that have been shown to be therapeutic in autism and other neurodevelopmental disorders that also can normalize some of the gene targets that we found, and systematically go through to see if these will normalize the respiratory rates of the cells and normalize gene function. And so far in the fibroblasts, we've done rapamycin, metformin, NCAR we're doing right now, and we have several others that we will do in the near future. The next study is very exciting, that we were very lucky that the Brain Foundation supported this. We have the very unique opportunity, since we are in a hospital setting, to actually have access to fresh excised brain tissue in order to look at this tissue and measure mitochondrial function in fresh human brain tissue. And this has never been done before. So of course, this has been done in animals, in animal models, but never in humans. And so we have really a unique opportunity to look at mitochondrial function in brain tissue, not just theoretically or through imaging, but actually using the seahorse instrument. And so, of course, the individuals that have brain removed have that for a reason, and usually it's for epilepsy. And we have children with epilepsy who do and do not have autism. So we can actually look at mitochondrial dysfunction, the changes associated with epilepsy, changes associated with autism, and correlating these with other biomarkers like high frequency oscillations, which we believe may be linked to a unique type of mitochondrial dysfunction. So we're characterizing mitochondrial function in those with focal seizures and other brain tissue. We also can correlate this with electrophysiological measurements and genetic changes, RNA expression, and transcriptomics, and also correlate this with high frequency oscillations. At this point, we've actually received tissue from different parts of the cortex, including the temporal, frontal, and occipital lobes, deep brain structures, including the hippocampus, parahippocampus, amygdala, and various pathologies, including cortical dysplasias and actually tumors too. So we are looking at some cancers. We've adapted a protocol that's developed by one of the scientists in our lab who developed this on animals, on rodents, when he was at University of Kentucky. And it not only allows us to isolate mitochondria, but also to divide into what we call synaptic and non-synaptic samples. We've adapted the seahorse protocol so that we can measure various aspects of the mitochondria and its respiratory rates. I'm not going to go into the details. Again, measuring both synaptic, non-synaptic mitochondrial function, doing genetic analysis, looking at mitochondrial morphology using electron microscopy, and also being able to localize these things in the brain. This is a picture of these high frequency oscillations, which we believe may be specific to seizure focus. And we believe that in order to have those high frequencies and mount neuron or energized neurons to have these high frequencies, you need very high respiratory rates in the mitochondria. So we're able to correlate this with this high frequency oscillations by depth electrodes, which we see here, also with MEG, and we also do resting state functional MRI and SPECT on many of these patients. And these are just some of the results. What we did find indeed was that the seizure focus had both complex 1 and complex 2 respiratory rates that were much higher than the non-focal part of the tissue, the part of the tissue that was not the epilepsy focus. So using that as a control, showing that the epilepsy focus had very high respiratory rates. And we were able to see the difference between synaptic and non-synaptic mitochondria, and finding that the difference tends to be in maximal respiration. We're also coordinating and collaborating with NIH and their electron microscopy lab at the Cancer Institute to also look at changes in mitochondrial morphology with electron microscopy. And here's a nice shot where we can see that they're the border of the seizure zone, the mitochondria on the left, is a little bit more intact than the mitochondria on the right, which is in the seizure zone, which is very swollen. The cristae, those folds, you can see are very swollen and very enlarged in those mitochondria on the right. So with that, I thank you for your attention, and I will take any questions.
There are questions in the Q&A section.
Okay. Super. So which is therapy which resolves this problem lifelong? Possibly. I mean, of course, we always try and use therapies that are going to be very safe and well tolerated. So they could be, if they needed to be lifelong or for prolonged periods of time, they could be. But the great thing about the mitochondria and what we hope is that the mitochondria is what we call plastic. There's what we call mitoplasticity. And we think that it may have gotten into this abnormal state because it is trying to account for certain stressors. And if we take away those stressors, maybe the mitochondria will help repair itself, and you can decrease the treatments. Can these gene regulation tests be conducted outside of studies? Well, not, most of gene regulation tests are really in the research phases right now. There are, definitely labs that are looking into how to use them as biomarkers. So I think that's in the future. So I don't think that we have those right now as more diagnostic tests. Yes, is the teeth research with Dr. Arona? Yes, most definitely. He processed the teeth for us. We published one paper in Translational Psychiatry, which was extremely interesting. I didn't go into it in detail, but the very interesting thing is that we found that the metals that were important prenatally were more nutritional metals of zinc, manganese, and copper, all which point to superoxide dismutase and control of oxidative stress, we believe, prenatally. But, we need to do more research, and we're trying to get NIH funding to continue that research because, of course, he's done some great research showing that these metals are dysregulated, and we're hoping to take it to the next step to actually show, well, what's the consequence of that prenatal dysregulation of metals. Regressive autism daughter, 16 years old, nonverbal, 5 mitochondrial markers, looks heavy mitochondrial impact. So yes, I mean, definitely the next step would be to actually measure mitochondrial function either with the buccal swab or other techniques, skin biopsy, muscle biopsy, if appropriate. Of course, you need to consult an expert in that field, in the field of mitochondrial function, to decide that, and also genetic testing, whole genome with mitochondrial DNA sequencing, probably. But of course, I'm not giving medical advice. You need to consult an expert, but I would pursue that. Did the kids with worse repetitive behaviors have high? Yes, they did. That's what we found in the lymphoblastoid cell lines that was from the AGREE database. I'm sorry, biorepository. Can these, which there is, if a child has consistently low numbers of mitochondrial swab, do they fall under? No, probably not. I mean, I'd have to look at that, but right, we see that there seems to be 2. I don't want to, we're very excited about these kids with very high respiratory rates because it's unique, but there's many kids with very low respiratory rates that fall into that category of more classic mitochondrial dysfunction who also need aggressive treatment. So so definitely important to pursue that with an expert in the field. Can this help with PANS pandas? Yes, most definitely. One of the things, of course, it's important to point out is PANS is not defined as an immune disorder. It is an acute onset of symptoms, and this, at least I'll tell you, when I was in residency, anybody with neurodevelopmental regression or changes, acute changes, had mitochondrial issues until otherwise, found. So I think that when we look at any acute changes in behavior, that we have to think in a very broad sense, and this is a whole nother talk I have of, of looking at the differential diagnosis, including, metabolic immune disorders, seizure disorders, and such. And we have to really cast a wide net when we look at those patients that we put in that kind of PANDAS category. And mitochondrial dysfunction itself can contribute to immune dysfunction. So at least in my practice, I found that a lot of kids, even with immune dysfunction causing PANDAS, also have a mitochondrial dysfunction, which needs to be treated simultaneously. So it's very complex. Is there a theoretical role for preconception and prenatal mitochondrial testing and supplementation? Most definitely. Most definitely. Is using lactate and pyruvate ratios as a screening tool not be enough? Yes, it's not enough. So lactate and pyruvate are just one measurement. One of the measurements that we find that that picks up some of these kiddos, lactate and pyruvate can be normal, but alanine to lysine ratio, which was really developed by Richard Kelly, is important. And of course, we have a paper on dysregulation of fatty acid oxidation pathways and unique changes in acylcarnitines that may be linked to mitochondrial dysfunction. So yes, it's complex. And especially, I think, in these new types of mitochondrial dysfunction, we're still learning what the biomarkers are. N-acetylcysteine, part of the neuroneeds mixture. I'm not sure. I think there is, but I would have to check the list for you. I don't want to misspeak, but the neuroneeds is a pretty good mixture, support mitochondrial function and oxidative stress, and I believe there's some N-acetylcysteine in there. If not, the only bad result on MitoSwab is borderline. What does this mean? Again, I think you need to consult an expert on mitochondrial function and looking at the MitoSwab to tell you that, because it can be tricky to sort out. Thank you for the presentation. Have you tested and identified ASD individuals who have normal mitochondrial function? Yeah, we wonder a lot of times, are there any normal mitochondrial function? Because we find so many. Yes, there are patients with normal mitochondrial function. Despite all the tests that we've done, there are a good number of kids that have normal mitochondrial function. So, when we can find it, it's good to treat. But it's not the only thing. I think it's very, the treatments are very complex, and they have to hit it from many directions. So when we can find that there's mitochondrial dysfunction, I think it's good to treat, but there's a lot of kids with normal mitochondrial function too.
Excuse me, Professor Frye, I think
yes.
you're out of time.
Okay, sorry, lots of questions. Great. So shall we move on?
Yeah, we'll put up the next bio.
Okay.
You can continue to ask.
Us all, for a long time. He's a research professor at SUNY Downstate in the Department of Medicine and Cell Biology. His research interests include biochemical and molecular aspects of B12 and folate absorption, transportation, and metabolism, genetic abnormalities in these pathways, and the cellular and metabolic consequences of B12 and folate deficiencies. He's looked at B12 and folate and homocysteine metabolism in elderly population with cardiovascular disease and cognitive disorders, including Alzheimer's, and looked at the neuropathology of these disorders and its effect on the brain. Active areas of research in his lab include, and are most important to autism, the folate receptor autoimmunity, how it relates to both neurotube defects and cerebral folate deficiency and autism. His research focuses on fetal and neonatal brain developments and the role of folate and B12 in this process. And he has really been the one that found the folate receptor alpha autoantibody, which I think is really an extremely important finding that will help us therapeutically in helping to improve the lives of children, both by prenatal treatments, improving neurotube defects, the development of or preventing the development of neurodevelopmental disorders, and autism. So he holds a bachelor's in chemistry from the University of Hunan, master's in applied biology from the University of Bombay, and a PhD in biochemistry from the University of London. So with that, I will leave it to Ed to give you a fantastic talk.
Thank you, Richard, for that great introduction. It's a pleasure to participate in this conference. Let me share my screen with you. Should I hit the full screen? Okay, good.
Yeah.
Okay. Right. I'm trying to see if I can. I'm seeing you, Richard. Can I see me?
I can see your slides.
No, but can I? Where am I? I don't see myself.
Oh, I don't know.
Okay.
I can't see you.
Okay,
But
all
I
right.
can see your slides.
I can see many people here. Okay. All right. So today I'm going to talk about folates, folate receptor autoantibodies, and the connection to autism spectrum disorders. In terms of disclosure, I would like to mention that the patent for the measurement of folate receptor autoantibodies is owned by the university, the research foundation, and I'm going to talk a little bit about the off-label use of folate or folinic acid in this case. So what is autism? We know autism as a behavioral disorder, and it is pretty evident that it is as a result of disordered brain development during fetal life and early childhood. The causes of autism remain unresolved. Consensus dictates that it is a multifactorial disease. That means it is not a single gene disorder. It does not follow Mendelian inheritance, and it doesn't follow typical Mendelian genetics. It is multifactorial, and those factors remain unresolved. Now, what is folate, and how does it connect to brain development and function? This is what I'm going to discuss today, and ask a rather reactionary question, like, could a single nutrient contribute to the clinical phenotype of autism? Okay. Now, just to introduce folate to the audience here, folate is a generic term used for vitamin B9. Folic acid, as we know, seen in preparations of multivitamins and other B-complex vitamins, is a pharmaceutical preparation that does not exist in the body, but can be readily converted to metabolically active form. It's used in pharmaceutical preparation because it's a very stable form and it can be readily synthesized. Now, normally during absorption, folic acid is rapidly converted to methylfolate due to the high dihydrofolate reductase and methylene tetrahydrofolate reductase activity in the gut. So at physiologic concentrations, when folate is introduced in the diet or orally, most of it will be converted to methylfolate before it reaches to circulation. Therefore, methylfolate is the major circulating form of folate and is taken into cells by the folate receptor alpha or the reduced folate carrier. Under physiologic conditions, it is primarily the reduced folate, the folate receptor alpha that functions, which has a very high affinity both for oxidized form as folic acid or as a reduced form of methylfolate. The reduced folate carrier, on the other hand, does not transport folic acid, but it can transport methylfolate and other reduced forms of folate. However, it needs a high local concentration for it to be activated and transport. All of the metabolically active forms are generated from methylfolate via the enzyme methionine synthase, which is a B12-dependent enzyme, and the dihydrofolate reductase enzyme. Okay, so before you get overwhelmed by this, let me simplify this diagram for you. What it shows is basically all of the single carbon exchange reactions and metabolic pathways involving folate. So folic acid is converted by dihydrofolate reductase to tetrahydrofolate, and that tetrahydrofolate pool then forms all of the reduced forms of folate that are actively involved in transporting single carbon units or carbon skeletons for the synthesis of various compounds. Among them, most important is the synthesis of purines and pyrimidines, which contribute to DNA and RNA synthesis and ultimately to cell replication. It is also involved in production of methionine via the homocysteine methionine conversion, and this requires vitamin B12. And the methionine formed is an important precursor for maintaining intracellular SAM, or S-adenosylmethionine, which is considered a universal methyl donor for methylation of DNA, RNA, and proteins. Okay. The homocysteine there is also a contributor to cystathionine production, which is very high in the brain, by the way. And cysteine produced from there is a source of cysteine for glutathione production, which is important for maintaining the so-called cellular redox state and mitochondrial function. On the other hand, if you look on the left bottom side here, one of the purines produces GTP. GTP is hydrolyzed by the enzyme GTP cyclohydrolase to produce tetrahydrobiopterin, which is the important cofactor for the hydroxylation of aromatic amino acids to produce neurotransmitters. So as you can see from this diagram, that there are so many pathways and so many reactions that can basically control cell replication and metabolic reactions. So it could have a profound effect on brain development and function, as you see in fetal as well as neonatal brain development. So to summarize, since folate has a major role in purine synthesis that leads to DNA synthesis and methylation reactions that leads to regulation of gene expression, this could profoundly affect neural development, and that in turn could affect brain function. And hence the connection between folate and developmental disorders. So looking at folate, there are many ways one could develop folate deficiency, whether it is malabsorption, low dietary folate, or genetic defects of folate pathways. And more recently added to this repertoire is the folate receptor autoantibody that could impede the function of folate transport. Folate deficiency, on the one hand, as seen on the left here, could lead to various pregnancy-related disorders. That includes infertility, miscarriage, preterm birth, neural tube defect pregnancy, and other gestational disorders. Shown on the right-hand side here is what could potentially happen after birth to the developing brain. Here you could have effect on metabolic processes, DNA RNA synthesis, again, the methylation and epigenetic regulation, effect on neurotransmitters, neurogenesis. Among them, what is seen and identified in autism is the cortical layering defects, axonal connections, and dendritic branching and pruning. So one can envision a scenario where basically so many different things are affected that not one single pathway or cause could be attributed to what we see in autism spectrum disorders. So just to introduce folate receptor autoimmune disorder here. Folate receptor autoimmune disorder presents with blocking and binding antibodies in the blood, which can be readily measured by standardized assays. What we know so far is that approximately 70% of the children with autism spectrum disorder are positive for both either blocking, binding, or both of these antibodies. These antibodies are prevalent in families with ASD children and ASD patients. What we know so far is about 38% of the parents are also positive for these antibodies. What is really interesting and fascinating is that these antibodies are also prevalent in unaffected siblings. Now, the main question here is, why are these children not affected? And this is a major topic for discussion, which I would not go into here, but just to indicate that there could be multiple factors that protect one fetus from the effects and another fetus is affected. So this could happen under various circumstances, and we could discuss this at a different conference later. Now, the presence of folate receptor antibodies causes these neurologic deficits associated with autism spectrum disorder. But how severe the neurologic defect is and the onset of the defect is dictated by the appearance and the titer of antibodies. So to show here, if at a very young age the child develops antibodies, this could lead to a very severe form of developmental deficit, such as the CFD syndrome. As the child develops antibodies later on between years 2 and 5, it could lead to spastic ataxic syndrome, intellectual disability, as well as learning deficits. And if the child develops later on between the age of 5 and 12, it could lead to milder ASD conditions such as Asperger's syndrome or psychotic disorder. And typically in older children, in addition to autism, we also find dystonia and schizophrenia associated with folate receptor autoantibodies. Now, in terms of the effect of antibodies, there are a number of factors that dictate the outcome. And here I'm showing you a diagram of maternal and paternal antibodies. If both parents are positive for the antibodies and the fetus is exposed to the antibody during embryonic and fetal development, the prognosis is poor and the disease is severe. If the father is negative for the antibody and the mother is positive for the antibody, again, this could lead to severe autism because the fetus is constantly exposed to the maternal antibody. And the response to treatment depends on how the early diagnosis and rigorous treatment. If the father is positive and the mother is negative for the antibody, typically this could result in moderate to severe autism with variable prognosis. So therefore, identifying parents and screening them for folate receptor antibodies becomes critical in the prevention and management of autism in the offspring. Now, what if both parents are negative for the antibodies? And this scenario does exist where the child develops antibodies to the folate receptor. And what we do know is the earlier the child is exposed to milk antigen, folate receptor is present in high quantities in animal milk and milk products. So the earlier the child is introduced to bovine milk, and if the child is susceptible to the autoimmune disorder, the child will develop high titers of the both blocking and binding type of antibodies and will develop severe CSF folate deficiency and cerebral folate deficiency syndrome. Okay. Now, if the child is exposed to antibodies, again, milk products early on, that child may not develop high titer of antibodies. It may develop a moderate titer of antibodies and will eventually show up as acquired infantile autism. Again, early diagnosis and treatment has a favorable outcome here. Now, the best scenario is when the child is not exposed to bovine or other sources of animal milk for a longer period, such as a mother breastfeeding perhaps for a year or longer. And in this case, the child develops antibody later on. And if at all, the child could develop a milder form of autism later on in life. So the best scenario here is to try and avoid animal milk and milk products as long as possible to prevent this sort of thing occurring. So in terms of treatment strategy, clearly a milk-free diet and high-dose folate administration would be the ideal thing to do. So the question here is now, what is the folate form of choice? And as pharmaceutical preparations, you have folic acid available, which is a very stable form, readily available and cheap. Then there is methylfolate, which is not so stable, but it is also readily available. And then there is folinic acid. The question is, which form should be used in the treatment of ASD? So to address this question, we looked at folate absorption in the rat. We took adult rats and looked at folate absorption after oral administration. So we used folic acid, 5-methyl, leucovorin, which is DL-folinic acid, only the L form is metabolically active, and fusilib or levoleucovorin, which is only the L form and is fully metabolically active. If you look at the purple bar down the panel, you'll see that no matter which form is used, typical peak activity concentration in the blood is seen around 60 minutes. The green bar shows conversion to methylfolate, and most forms are converted to methylfolate, perhaps more so in the case of levoleucovorin. Also, there is more tetrahydrofolate formed from levoleucovorin. The one thing about using folic acid is that, as you see on the red bar here, there is a significant amount of unconverted folic acid appearing in the circulation. So looking at unconverted compound appearing in circulation, the red bars show folic acid, and there is a very high amount of folic acid in circulation. Mind you, the dose administered here was 4 milligrams per kilogram body weight, which is twice the dose that is highest dose given to children with the autism spectrum disorder. And at this pharmacologic dose, clearly there is a significant amount of unconverted folic acid appearing in circulation. There is some recent literature criticizing the use of folic acid even as a supplement because they feel that the unconverted folic acid has a negative effect metabolically, and it may be detrimental to folate metabolism. So clearly, in this case, where pharmacologic doses are administered, folic acid is not the compound of choice. Methylfolate, on the other hand, can be administered, and it appears unconverted into circulation, as seen here, and it can be readily converted to other forms of folate via the enzyme, the methionine synthase and dihydrofolate reductase. So looking at the next two panels here, D-L-folinic acid and levofolinate, most of the levofolinate is converted to methylfolate. And the advantage of using D-L-folinate or levofolinate is that both forms can be used. Both forms can be utilized in metabolic processes. So even if it is not converted to methylfolate, the form that is not converted can be readily used in metabolic reactions. So to connect folate receptor antibodies to autism spectrum disorder, we looked at the rat model of exposure to antibodies during pregnancy. A rat model of exposure to folate receptor antibodies shows behavioral deficits such as social interaction deficits, learning, memory, and cognition deficits in the pups. And this data is all published, is out there in the public domain. These deficits can be prevented by pre-administration of folinic acid. So what could be the potential mechanism, both of the pathology as well as the rescue? To investigate this, we looked at folate and folate antibody transport to the fetus and to the brain. So shown on the right top panel here is the brown staining material is the presence of folate receptor antibodies, in this case, rat folate receptor antibody administered intraperitoneally, and its distribution in a pregnant rat. So you can see there is a tremendous accumulation in the placenta, in the yolk sac, as well as in the embryo, much less in the embryo. A quantitative evaluation of that indicates that most of the antibody collects in the uterus, and in the placenta, very little goes to the embryo. And you can see higher magnifications of the embryo here, again, accumulation in the embryo as well as in the chamber ventricles and the choroid plexus. Now, what is the result of that? The result of that is if you give now radioactive folic acid to an animal which has received the antibody, what it does is it blocks folate. It blocks folate transport. That blockage is seen most in the embryo. Here's the normal embryo. Here is the embryo receiving folate receptor antibody, 1 or 2 doses, depending on 48 or 72 hours. And that results in a significant decrease in folate transport to the embryo. Let me get this thing out of there. Okay. All right. Next slide. So then we asked the question, is the distribution of the folate itself, in this case, we use biotin conjugated folic acid to try and trace it. And when we gave it to pregnant rats, as you look at the distribution in the placenta and in the embryo itself, this is as early as 30 minutes after IP injection. You can see there is tremendous accumulation of folic acid in the placenta, in the embryo. You see at 30 minutes, there's a lot of accumulation in the heart, in the liver, and also regions of the brain, but much less so. And by 1 hour, there is more accumulation of this in the embryo, and a higher magnification down here shows all that brown staining stuff is now folate distribution within the uterus, the placenta, as well as the embryonic tissue, including the choroid plexus and regions of the brain. So clearly, the antibody distributes to regions similar to where the folate would be distributed, and this could affect folate uptake and deposition within the brain tissue. And when we looked at a young brain, in this case PND22 rat brain, again IP-injected biotin PGA distributes to, this is the cerebrum, this is the cerebellum, and the time course ranging from 5 minutes all the way to overnight. And as you can see, as early as 5 minutes, you start seeing the appearance of biotin PGA within the brain, much less so in the cerebellum, but with increasing time, and by 30 minutes, you see a fairly large accumulation of folic acid in the cerebrum as well as in the cerebellum. And this accumulation, even though it seems to decrease a little over overnight period, it seems to remain so much so in the cerebrum. And the distribution is typically seen around the tracts of the white matter in here. And seen in the next slide is a higher magnification of this, showing you distribution of biotin PGA after intraperitoneal injection. As you can see, most of the distribution is in the white matter tracts, in the internal capsule area, the external capsule area, the fimbriae, and in the ventricular chamber in the choroid plexus. Now, if the folate is administered 24 hours after antibody administration, this distribution is decreased. You can see there's no specific area of decrease, but overall there is a decrease in folate distribution within the white matter of the cerebrum. And if one looks at the antibody distribution, you can see there is a high accumulation of antibody in the choroid plexus. Antibody distribution can also be seen in the microvascular, in the blood vessel, in the brain itself. So there is a clear effect of the antibody on folate distribution in the brain. And this could be the reason why the presence of antibody leads to cerebral folate deficiency. Now, one of the questions we have tried to address is the behavioral deficits that we see in pups exposed to antibody during pregnancy, is this transmissible to subsequent generation? If it is, it would indicate that it is epigenetic. There are epigenetic changes. This could be in the methylome, gene expression profiles, and then it could readily be transmitted to subsequent generation. The reason for asking this question is that autism runs in families. But it is not a genetic disorder in the sense that Mendelian genetics is transmitted, but it is a disorder that runs in families. There is a genetic component involved. So we wanted to see if the behavioral deficits that we see in F1 generation, these are the pups exposed to the antibody during pregnancy, would transmit to subsequent generation. And from this preliminary study, whether it is looking at vocalizations on PND4, which is an indicator of bonding between the mother and the newborn pups, or whether it is looking at male vocalizing of bonding in older pups, as well as active and conflict avoidance tests, which are indicators of deficits in higher learning, such as learning, memory, and cognition. Are these present in the first generation, and are they transmitted to subsequent generation? And from this experiment, it appears the answer is yes, that in fact, the deficits that we see in the pups directly exposed to the antibody are transmitted to subsequent generation. So what is the relevance of this experiment? And how could this experiment then lead to subsequently identifying the potential causes of developmental deficits that we see in patients exposed to folate receptor antibodies lies in the fact that this animal model allows us to look at the epigenome, the methylome, and also identify the genes involved. It also allows us to basically experimentally manipulate the epigenome and to see if we can change it or restore it so that we can prevent these deficits being transmitted to subsequent generation. These are very important studies that could perhaps provide an answer to the behavioral deficits that we see in exposure to folate receptor antibody and folate deficiency. We are most grateful for the Brain Foundation for funding this ongoing research, and hopefully for the future research, which is going to need a lot of funding to really look at the epigenome, the genome, and to see how we can restore the normalcy to this genome to prevent transmission of these behavioral deficits. So in conclusion, levofolinate has shown great promise in the treatment of ASD. And this is evident from trials that have already been completed by Dr. Frye and Dr. Ray Makers. And with significant improvement in speech, communication, and social interaction. There are at least 3 clinical trials ongoing, and hopefully this will give us more data. I believe that it holds most promise, perhaps in the prevention of ASD by identifying and treating parents prior to conceiving. Therefore, screening for folate receptor antibodies, reducing antibody titer, and providing adequate folate are critical for a positive outcome. Maternal cord blood testing for folate receptor antibodies could provide for follow-up and early intervention in the newborn to prevent development of ASD. So that's our ultimate hope, to see how we can prevent the development of ASD. The last data I heard from the CDC is now 1 in 44 children diagnosed with ASD. This upward trajectory needs to be changed, stopped, or shifted to the downward trajectory. And our hope is that we may be able to do it if we can identify what is the cause at the genetic level that is causing this. I have many people to thank for some of the work that I presented here today. Up top panel is my lab group. Most of the work I presented was done by an MD PhD student, Ankuri here, and Jeffrey Sequeira, my research assistant. Current ongoing work. Oh, how do I get back to this? Jeez, what happened? Okay, current ongoing work is being done by Natasha, my graduate student, and hopefully we will have some answers in the near future. Thank you for your attention.
Thanks so much, Ed. That was really terrific. What a wonderful talk, and it's such promising work. I think Dr. Woody has the first question.
Okay, how do I get back on? I can't see myself, or?
Just stop sharing.
Stop sharing. Let's go back. Okay, great. All right, I'm back. I'm sorry about that last slide. It flipped and I went back to the first slide. Okay, so the questions are in the QA or chat?
They are. Dr. Woody had the first question. Did you want to?
Dr. Woody is in chat or Q&A?
No, he can just say it if he wants.
Oh, okay, sure.
If not, you can start.
So, Dr. Woody, you have a question?
Yes, I do. Are you familiar with the new therapy using anti-FcRn antibodies to eliminate pathologic antibodies in patients, which has actually been used in pregnant women to eliminate anti-red cell antibodies?
Yeah, there are treatments to eliminate antibodies. There are also treatments in using drugs that reduce antibody production. There are also treatments that use IgG, infuse IgG to reduce antibody. These are all general treatments used for a variety of autoimmune disorder. They're not specific for autism related, but they have been used in CFD patients with severe CFD syndrome, where the antibody titers are very high. I'm not aware of anything that has been used to specifically remove folate receptor antibodies.
Yeah, the rest of the questions are in the chat. Do you have the chat, or you want me
Okay,
to read them
yeah.
to you?
Would you read them to me, Richard?
Okay.
That would be most helpful.
So the first question is about, the study that showed high doses or high levels of folic acid in the blood maternally has been linked to autism. And the question is, how would a too high dose of folic acid in the pregnancy lead to increased autism? Would it be through in vitro hurting the child or through an autoimmune reaction of the mother?
Okay. Most of the studies reported, they did not involve pharmacologic folic acid administration. They were in the general population showing small amounts of unmetabolized folic acid. My explanation for that is if you're on a daily B complex or multivitamin that includes folic acid, you're likely to see some unmetabolized folic acid in circulation. There could be some unmetabolized folic acid intracellularly also because the enzyme dihydrofolate reductase is not able to very efficiently metabolize all of the folic acid. So if the concentration drops below the Km, you're going to have some folic acid in the circulation. Whether it causes autism, I'm a bit skeptical about that. I'm not sure. I'm not buying that theory, even though they have shown that there is some link statistically between these two.
Okay. Next question is, given the transgenerational influence, at what gestational stage is the FRAT most beneficial for prenatal testing?
Ideally, the FRAT should be used in parents before conceiving. If both parents are positive or one parent is positive, they should be treated both to reduce the antibody titer and to provide adequate folate. That would be the ideal scenario where the parents are screened prior to conceiving. And if that is not done, then I think introducing larger doses of folinic acid during pregnancy and also during early childhood may help prevent the disorder, those who are susceptible to the disorder.
Okay. Now, an interesting question. What is the range of epitopes? What is known about the immunogenicity of the bovine folate receptor, that is immunogenic polymorphisms? Any info on different strains of cows? Have you looked directly at the methylome in rats in high titer or in high titer humans?
We have not looked at the methylome in the rats or humans, but that is clearly our objective to going forward, to look at the epigenome in some detail and to see what changes have taken place and can we restore these changes by providing folinic acid to the rats before pregnancy and during pregnancy and see if the outcome changes. Now, what was the first part of the question?
Whether there's any epitopes specifically. Is
Okay.
there, do you know anything?
Yeah. So what we do know is that bovine milk is highly immunogenic. So is camel milk folate receptor. The least immunogenic is actually goat milk, even though goat milk contains about twice as much folate receptor as bovine and camel milk. Okay. Epitope-wise, the antibodies that we have identified are blocking type. So they bind to the active site of the folate receptor, whether it is irrespective of which source the antigen comes from. And then there are binding antibodies which identify epitopes that are distinct from the folate binding sites. All of these antigens have greater than 95% homology in terms of amino acid sequence.
Okay. And one more question we have time for. Is there any relation between maternal age and FRAT?
Maternal age and FRAT. Good question. I don't know the answer to that because we really haven't looked at age-wise. I can tell you this, that when we've looked at women with neural tube defect pregnancy, these women mostly were in their late 30s and 40s. they've had an NTD child maybe 8 years earlier or 10 years earlier, but they still were positive for the antibody. So once you have the autoimmune disorder, you're likely to be positive for a long time. Now, one thing that reduces the titer of the antibody and eventually gets you to negative area is if you go on a totally milk-free diet, as was the case with one mother who, we have published this, had repeated miscarriages and had difficulty in conceiving. She had a high titer of the antibody, and she was put in a milk-free diet for over a year. And at the same time, she was monitored for this antibody titer. And eventually, she became negative for the titer. She conceived normally, carried to term, and had a normal child.
Super. Well, you can answer the rest of them in the chat, Ed.
Okay.
You can get to it, and we can move on to our next speaker.
Thank you.
Thank you, Ed. All right, so it's my pleasure now to introduce Mentioned Child Find program. He's been the PI of several NIH and NBHB funded studies of interventions to improve early development. He's the head of the LEND program at SUNY, and he has many interests. And with that, I will let him give you great information about his work on folate antibody and trajectories of autism in early childhood identification and treatment. Do you have your PowerPoint? Yeah, great.
Sorry, can you all hear me? And
yeah, just from the beginning. Excellent.
Okay. So what I'd like to do is to go over very, very quickly, because I think Ed has covered it, an overview of folate receptor autoantibodies, the linked ASD, use of leucovorin. I'd like to show two clips of a before and after of a child we treated, a 4-year-old, with leucovorin, and then look at an objective measure, the BOSC. I'll talk about that in a minute, to look at how you objectively measure social communication change. I'd like to come back to the questions that Ed raised about postnatal and prenatal impacts, and then finally move to how this is the rationale for our current Brain Foundation funded study. So let me start. Just to say, I have no financial interest in any of these. We will talk about non-FDA approved use of leucovorin. So can I just say, as a pediatrician, I've worked, our clinic largely sees children on the spectrum at different ages, and we worked for a long time. We see it as a complex, multifactorial process. So when a colleague said, introduced me to some of this work, and Ed's work and Richard's, that, oh, autism, it's due to a lack of folate in the CNS, I must admit that my reaction was this. It sounded like something out of a tabloid. It sounded like, oh my goodness, autism cure found and it's a vitamin. And I was skeptical, to say the least. I've come around. If I could just go over, there's some very convincing evidence on this, that folate receptors disrupt the folate receptor alpha, that it results in CNS folate deficiency, the strong link to autism spectrum, that you certainly see a very, very high rate of, in children diagnosed with ASD, this would be comparing to folate receptor positive rates, maybe of 10 to 15% in the general population, depending on the study. But most strikingly, and this is especially work that Dr. Frye has done, that treating with leucovorin, bypassing the folate receptor blocking step and correcting central nervous system folate, really shows a clear clinical improvement, certainly in language, but also in social communication in preschool and school-age children. So let me start by seeing if I can get this to work. I'm going to show a 4-year-old. Sorry, if I could just go back. So this is a 4-year-old with autism spectrum. He had language delay, very poor social interaction, and we arranged the testing through Ed's lab and he was positive for blocking FRA. So let me show, if I just open this.
We have more race cars. Let's open this. Let's see what we can do with this.
Can I just ask, are you all able to hear that? Was that okay? Was that working?
Yes.
Yes. Yep.
Ooh, that's so cool. One for you. Look, and one for me. Should we race the cars? Let's see what this does. Put this down. Oh, good job. Can you turn this? You turn it. Can you turn it? Yes, good. What is that? What animal is that? That's a chicken? Good job. What else do we have? Ooh, another one. Ooh, let's do it together, Nikos. Let's see, can we do it together? You do one and I'll do this one. Look, which one do you want to do first? You pick one here and I'll do the same one here. Okay, you want to pick this one? Oh, you want to do race cars? Ready? Whee! Look. Look at this. Look. Ready? Should we make it go? Pull it back. There it goes. There goes Nico. Look. There it goes. Oh, there it goes. You want to make yours go? Here, put your guy in here. Pull it back. Here, look. Pull it back. And go. Didn't quite go. Here we go. Ready? 1, 2, 3. Go! What else do we have in here? What is that?
He loves music, so those triangles will probably be right up his alley.
Oh, look! Dump truck. Do you want to put some blocks in it? Yeah, look. Do we put blocks in the dump truck? That's a dump truck. Let's put some blocks in it.
Okay, I think you get a flavor. Okay, so we treated him for 6 months with 2 milligrams per kilo of leucovorin, and this is when he came back.
Okay, I want Nikos. Look. Which one should we open? Can we do this together? Which one should I open? You want to open number 1? I have a chicken also. Is yours not closing? There we go. You turn that.
1, 2, 3.
Wonderful. Now you open yours. Same way.
What's this?
Turn that. What is that? What animal is that? A chicken, yes. Who's under 2? Who's that? What animal is that?
A chicken.
Under 2. Under number 2. What is that? What animal is that? Open that. That's a sheep. Open number 3. You got to turn it this way, like that. Turn it. Yep. Oh, I think you do. There you go. Who's that?
Movement.
What animal is that, Nico? This one. That's a horsey. That's a pig. Should we see what noises these animals make? Look. I have a farmer here. See? And where does the horse go? Can you tell me where these animals go? Tell me where these animals go. Look. There's a picture of the animal right here, and then you have to match the animal. Can you match the animal with the picture? I'll help you. What animal is this? Where does pig go? You see it?
Right there.
Yeah, that's right. And then it makes a noise. You want to push him down? Push him down. That's a cow. That's the farmer. That's a horse. Where's our sheep? Did we lose our sheep? Oh, here she is. There we go.
She's right there.
Yes. And if you press them down, you'll hear the noises they make.
That's pretty impressive, Harris. I think maybe we move on in the sake of time.
Yeah. Do you know the song? Oh, MacDonald had a farm. I'm not working. It's not working. So what should we do now?
Okay. So, so we used a measure called the BOSC, and this basically is a brief observation of social communication change. I don't want to go through the algorithm. The point is that for this and for several, about 15 different features, it really operationally has you score. It's derived from the ADOS by the same authors, with a low score being a more typical child, a higher score being more autistic. So on eye contact, this child went from baseline of 4 before and now scored a 1. On social overtures, he went from a baseline of 5 down to 3. If you look at the overall scoring, if you add these all up, he first of all, if I could just ask clinically, is this child still on the autism spectrum? I think the answer is yes. But with an overall score going from 39 down to 19, I think it's consistent with both the parents telling us and our own impression that this is a much more cooperative, engaged, and responsive child. Okay, so this led to proposals that we could ameliorate ASD by early identifying and treating it. But I think the treatment strategies, as Ed started talking about, will depend on understanding what's going on with FRA. This, as Ed covered, are we talking about sort of postnatal exposure to milk, for example, triggering some degree of titer and leading to this, and then we would look in the pediatric period to identify and treat to reduce and autism. On the other hand, if we're talking about parents who are positive and this is being transmitted, and especially when the mother is positive, then you want to look at screening mother and father and looking at obstetric care. Why does it matter? This is a work from Dr. Raymakers. I think he presented last year. Basically, this is the CARS score, where anything over 30 would be meeting threshold and the higher, more autistic. Comparing blue baseline, he had a range of kids of different ages, but then treated them for 2 years, and this would be the score after 2 years of leucovorin treatment. And where K is the positivity of the child and the mother, P the father. So if you could just look at the difference when the child is positive, but both parents are negative, the baseline, first of all, across is somewhat lower. The improvement with leucovorin much more striking. At the other end, if the child is positive, both parents are positive, they're starting out at a much higher baseline, and in fact, the improvement is slight and not significant. So this matters. As Ed said, FRA positivity runs in families. That something like 60 to 70 in one of his studies of parents as well as non-affected siblings were also FRA positive, raising the question, why is it that if you're FRA positive, why do you sometimes get a picture of ASD in some individuals and not in others? Okay, so I think the answer clearly has to do with timing of when the FRA is turning positive, as well as the interplay of many other factors, metabolic factors, genetic factors, and exposures. As far as I know, though, there have not been longitudinal studies. The studies we've heard so far are cross-sectional, looking at where a parent or a child is long after a child is diagnosed, but there haven't been studies starting at birth and looking forward of FRA status in the parents and the child in terms of their ASD development. And so the question we are asking in our Brain Foundation study is, if we were to examine the trajectory of FRA status and looking at later diagnosis of infants and starting with these are infants at risk for spectrum based on already having parents coming in with an older sibling who is positive, could we tease apart the contributions of FRAs that are present in the newborn, as in prenatally, from those that develop later in infancy, and how does that correlate with the emergence and severity of ASD features in subsequent years of life? Okay, so the goals of our Brain Foundation and O'Sullivan Foundation grant study, first of all, to look at that, to look at the trajectory of FRA status in cord blood, and then at 6 and 12 months, and compare that to the clinical course. We will try very hard to go through the second year of life. It's a short, the grant period is short, but to look at when and how much do symptoms and a diagnosis of autism emerge. Secondarily, to look at brain function, to use FNIRS, functional near-infrared spectroscopy, which is a non-invasive neuroimaging, as well as what's more classically been used is eye tracking, as early biomarkers to look for, even in infancy, and there's studies on certainly in eye tracking and some in FNIRS suggesting even before autism emerges, you can see differences in brain function, response to social stimuli versus not. And if we can then see how they correlate with FRA status and eventually a later ASD diagnosis, the overall goal would be to pave the way for studies using leucovorin treatment and in infancy and eventually prenatally. So how we're doing this, we'd like to enroll 30 to 35 pregnant mothers who have a previous child with autism spectrum, who are going to deliver at either downstate or some of our affiliated hospitals, to determine the status, FRA status in cord blood, we're figuring we're at each step going to get some loss to follow-up. Following this cohort through the first year of life to look at ultimate diagnosis with ASD, measuring FRA status at 6, 12, and 24 months, in addition to looking at a whole series of environmental factors, toxic exposures, as Dr. Frye talked about, and especially milk and bovine milk exposure. And then to look at their cognitive as well as social development at 6, 9, and 12. There are not a lot of measures in the first year of life. One is the communication symbolic behavior scales. The Mullen is used for cognitive and language. We would also use the FNIRS and eye tracking. And then the idea would be to continue them into the second year of life when you can start to use more classic gold standard measures such as the ADOS, the ADIR, the Autism Diagnostic Inventory, and continue with the. So in conclusion, if we could better understand the child's FRA trajectory, the ways it's influenced by the parents' FRA status, environmental factors, how these correlate with manifestations of ASD and brain function, could we then better design treatment and preventive interventions, including what kind of approach? Are we starting in infancy, or should we really need to start in prenatally, as Dr. Quadros? So getting back to is a vitamin going to cure autism? I think probably not. But is folate treatment, can it make a major difference in ameliorating and improving functioning in very large subsets of individuals with ASD? I think so. So thank you very much.
Thanks so much for that great presentation, Dr. Huberman. we're supposed to have a break. We're supposed to have a 15-minute break, but I would propose that if you could take any questions in the Q&A, through typing in, and maybe we take a 10-minute break and come back at 11:05.
Yes, that's
All right, that sounds great. Thank you, everybody.